Yes, I think you can record it. I mean, most of the work that I'm showing is published, and I will also be sharing some of my startup journey, but I think it's also, I hope it's interesting for general public. Yeah, in fact, I think it's first time that I'm introduced as industrialist. I used to be an academic for quite a long time. Actually, yeah, for last 15 years, PhD, postdoc, some research time at Max Planck in Mainz. And yeah, last year I have made a leap and started a startup, Phos4nova. It's a spin-out company of University of Twente. And yeah, I would like to take you with me on my journey, which is partly scientific, partly also my startup experience. I hope it will maybe motivate more young people to, or not only young people, but generally more researchers to go on this route. And yeah, why did I do it? I do it because yeah, I mean, I believe that science can solve bigger challenges, and particularly I'm in biomedical field, so I would like to create a world where one day no one will have to fear severe diagnoses such as cancer and Alzheimer's disease. And as a community, we are working on it already for a long time, but there are still several challenges. And yeah, we are here at Polymer Conference, and I think we all know that polymers have huge potential to cure diseases, for example, to deliver drug, to develop new regenerative tissues. And in fact, when I was a student, and that was back in Mainz, so Ringstorff University, I was thinking, okay, with this we can create sort of a magic bullet. However, if you look at some numbers, that's what the reality looks more or less like today. So these two numbers are what you find if you search on Web of Science, either polymer and drug, or if you do the same search on the database for clinical trials. And what it tells us is there is really a lot of excellent science going on, but only very small fraction of this science actually finds its way to the clinic. And yeah, the reason is that we actually inject our materials in the patient and we think they can do a lot of things, but in fact, we actually do not really know what happens with these materials in vivo. And the patient remains a black box. But at the same time, knowing what is going on is the key to develop efficient therapeutics. And we need to know quite a lot of things. So first of all, we want to know if we have, for example, nanoparticle, if it goes to the tumor, how much materials goes where, what are the properties, or especially for implants or for regenerative hydrogels, do these properties change? What happens with materials over time in the body? Is it still the same after 1 month or after 1 year? But so far, retrieving this information is very challenging. And the reason is because what we do now is we have our construction is usually polymer, or well, lipids are also widely used other materials. And when we want to trace it in vivo, we do attach a label, which is usually a contrast agent. Then we inject it. But then we only see the label. In fact, we have no idea what is happening with the functional part, because so far there is no way for direct monitoring of polymers. And what we want to create is actually a paradigm shift of this approach. We want to develop a polymer that can produce intrinsic imaging signals, so that instead of this situation, we go to this situation where we can monitor the polymer, tell if it's degrading, tell how much it's there. So far, well, nice concept. How can we actually do it? And that's where the challenges start, because polymers are very similar to biological tissues. And so in my team, we turn to MRI, because MRI is a very great technique. It's safe. It can provide all the information that you can get in NMR, and it's widely integrated in the clinics. So there are more than 95 million MRI scans done every year. But nowadays, MRI requires contrast agents, and that's where the problem starts, because currently, in the clinics, you use mostly contrast agents which are based on heavy metal gadolinium, or actually gadolinium ions, which first of all have performance issues, so they are highly sensitive, but they only change the contrast. So what you get is basically, 50 shades of gray, as you can see here on the left image. And also this heavy metal can accumulate in organs, so there is a lot of discussion in MR community about the toxicity concerns, and it also enters rivers, seas, and even found in drinking water in major European cities. There is another way, which is better in terms of sensitivity, and that's what is called heteronuclear MRI or hotspot MRI, where instead of detecting proton as it's done with normal MRI, you can detect other nuclei. And so far, the focus has been on fluorine mostly. So normally, like, for example, system I worked during my postdoc contain highly fluorinated compounds, PFAS, and PFAS are generally challenging to deal with. They are hydrophobic and lithophobic, so they are colloidally unstable, chemically extremely stable, and there are some ways to create polymers that are also labeled with fluorinated units. By the way, Sergey did excellent work on this, I believe. But still, at the same time, to make it stable, the fluorinated part is not the functional part, which deals encapsulation or targeting or any other biological function. So at the end, we still are dealing with a label. And in our case, we decided to turn the system around and came up with the idea of using phosphorus for imaging. So, well, by using phosphorus. In fact, the idea is at first, my MRI collaborator it was not a very smart idea because phosphorus 31P, it's a natural isotope, but there are also many phosphates in the body, so you get natural background. It has low sensitivity. And also polymers are in fact sort of too solid for MRI, so the magnetic resonance relaxation properties are not ideal for imaging and they are not suitable for conventional sequences. But luckily, my MRI collaborator had no clue about polymer chemistry. And what we decided to do was to use polyphosphoester chemistry to solve these issues. And polyphosphoesters, just a brief introduction to this polymer class. These are bio-inspired polymers where we have this phosphorus binding motif, similar to DNA and RNA, but can do chemistry on Z and can I actually have a pointer? Yeah, I can do chemistry here on these two groups. And so, yeah, my team was implemented in the team of Frederick Wurm, who already had a lot of expertise with these polymers, particularly for stealth application and PEG replacement. I will come to this at the very end of my talk, but also as therapeutics. But yeah, those were phosphates and in fact also not really suitable for imaging. So what we developed eventually was a new concept how to make them traceable. First of all, to overcome the background, we can change the chemical shift. So we move from phosphates to phosphonates. We can also play with the concentration of P31 and form colloids. And yeah, luckily polymers also have a glass transition temperature. So if we move to lower TG, we also can further tune the relaxation times. And yeah, the final result, it was a long journey with several failures at the beginning, but the final result was this gradient copolymer structure, which can obtain by copolymerization based on kinetics, as was shown also by kinetic studies in Monte Carlo, and this polymer self-assemble into micelles, which are in this long circulating micelle regime. And yeah, once we had it, we also confirmed that they have good MRI properties in terms of relaxation times and pitch. And then we tested our first patient, which was a while. So it was a couple of weeks after my MRI collaborator told me, "You are crazy." We had first sample and then we put it in the MRI machine. And here you see the cuts to the vial. So that's really MRI, not spectroscopy, but image taken with spin echo. And yeah, after that, we moved on with testing the systems in vivo. And yeah, I used these animals. It was collaboration with Phos4nova in Germany, who is working on developing this caterpillars as alternative animal model. So if anyone wants to have a connection here, I can also provide you a contact. Basically, they have more or less same size as mouse and are very easy to handle. And yeah, what we obtained was indeed, we confirmed that we could detect these materials in vivo, that there is no background. And yeah, after 24 hours, we still saw that they're circulating here in the hemolymph, so it's caterpillar blood. So it's something what is expected for micelles in this size regime. And yeah, also the signal is quantitative. So That's again now a vial experiment. So it's really linear relationship as same as for enema. And yeah, finally, we could also show that we can monitor the property change. So here we injected the materials in the caterpillar's gut, and what the animals are doing most of the time is eating and, let's say, getting rid of their food. So we collected the feces, resuspended them, and ran a literally shitty NMI experiment. So we took a sample of feces, and here we can clearly detect the degradation products of this polymers. And yeah, so with this, we can tell where is the material, we can quantify it, and we have also the first hint that we can in fact look into the properties change in vivo. And yeah, so far so good. With these results, we also did the first proof of concept for drug encapsulation, where we encapsulated hydrophobic drug product and showed that indeed it's stabilized by mice cells and reduces the viability of cancer cells. I mean, it was back then a very small study, so it's something we still want to expand on. But yeah, at the end, what we have created is a platform technology of this MRI traceable polymers that could be used either as MRI agent or also show potential for applications in nanomedicine or in biomedical materials. And by the way, we are looking now for partners and collaborators who like to work more into this direction, who have, for example, biomaterial expertise. So if you are interested looking into this, please reach out to me, drop me an email. I have my contact at the end. So far so good, but now I would like to come to the second part of my talk, because as I mentioned in the last time, I transitioned from academia to becoming a startup founder a bit more than a year ago. And yeah, it was together with a team with Frederick Wurm and with Timo, who was back then still completed. No, back then he already graduated, but he completed his PhD on this topic. And yeah, how did all this go? Well, when we had the results, we started discussing with the University of Twente if we want to file for a patent, and that's where it started. So Twente is very entrepreneurial university, equipped with incubator and all the things. And when we first mentioned patents, I got in touch with business development. Then, well, we had to think what we actually do with the patent and how we want to monetize it and how it should all go. And that's how it started. So we joined the incubator, started the workshop, got some pre-seed funding to do a little bit of customer validation. It sounds easy. It was actually, yeah, quite a lot of work because, yeah, everything was relatively new. By the way, it still is. It will not perhaps change in the next 5 years. But that's how we eventually started out, by going to the incubator and thinking of how we can do it. And yeah, when you go to such incubator, they will do you to verify your product and actually find things like who would buy it, who would maybe invest in it, is the production scalable, etc., etc. When you're doing it for biomedical product, you will realize that you actually have a very complex technology roadmap. Your buyer is not essentially your user, not essentially the person who is going to benefit from the product, because you have this network of medical doctors, insurances, and all those things. So it's a relatively complex puzzle, which we are still putting together, but that's how we started. We did some market analysis. We had a look, okay, there is a market, and then, yeah, maybe also a bit unprepared, we went into starting, let's say, a company and figuring out how to compare. So now our current business model is actually marketed as a patent and as a platform technology for different applications, and focus ourselves on clinical MRI market as a startup for first clinical market entry. But again, as I mentioned, there is a lot of more potential. And yeah, but actually how it went all the time, we had some evolution of our business model, because first we thought we will do contrast agent. Then we went to polymer supplier, then the contrast agent, and then again, like over this 1.5 years, or actually, well, 3 to 4 years now, it evolved quite a lot until we ended up with doing a platform. So what happened in between was, well, we talked to a few clinicians, had some interaction with CDMO, decided, okay, maybe contrast agent is too complicated, let's do a polymer supplier. Then we had some first exposure to investors who were really interested in contrast agents. So we decided to go for contrast agent, and yeah, eventually with more investor exposure and startup competition here in the Netherlands. We ended up with doing a polymer platform. By the way, if you consider starting a company, I recommend doing getting involved in academic startup competitions, because then you get into the infrastructure, you get to talk to people who have similar struggle, and that is really helpful. And so that's how it went out. Now we are 1.5 years into the business, and we are progressing. I always say that it goes like this, but the overall direction is up. Let's see where we will end up with this at the end. But yeah, maybe also several things to keep in mind for this very early starting phase. So first of all, plan negotiation with your institute if you go on it in advance, because deciding on IP transfer, term sheet, and all these things, it will take you at least half a year. For us, it was, I think, more than half a year. And at the same time, you should be aware for deadlines, because there are some, and that varies from country to country, national fundings for startups, which all have deadlines. So ideally, if you want to apply for something as a startup that year, you should be incorporated already before you apply. And get ready to be jack of all trades, because I think the first months after starting a company was the most chaotic months I ever had. Forget PhD examination, forget the final exams, because you have to be jack of all trades, and you deal with things you never done before, like opening company bank account can be complicated, and particularly with the companies in the Netherlands. I live in Germany, which is well, very, very close. We're in the eastern part, but anyhow, in the EU, it's still sometimes problematic. You need web page logo, maybe bank protection, and then there will be for sure some other bureaucratic things you maybe have even no clue about what it is. So the very early stage, I mean, it's all doable. There are many people who already handled it. I also obviously survived, but just get ready for the chaos. It will get better at some point, so luckily. Yeah, and then also I think my take home so far about how it went. Yeah, the most important thing is you have to enjoy the ride. So especially like maybe it's advice to PhD students who are thinking of it. Yeah, you should figure out what you like doing, and if you like to go on this journey and see how to do it. So you have to like your product, you have to also like this, different type of work, I would say. Yeah, you will, exactly. So that's already here. And if you have a partner, make sure that he or she supports you. It is usually really helpful that you have him on board, maybe also your friends on board, so that you have some personal backup where you can go and maybe complain when the things do not go right, or play as a team. I mean, you all together, so celebrate, take time, and there will be conflict situations, even if you know yourself for years, which is the case for us. It's not always perfect, but you need to find a way how to roll potential issues out and how to support each other. And build a network, because especially as a woman in tech, I must say that it's still very male-dominated world. So there are only relatively few startups founded by women. I mean, luckily there are now some European networks and other networks, but it's maybe here at the technical university it is like this, but I think overall it is still important that you have your network and your support network. And last thing that I want to announce, we are now actually, after all this chaos, we are preparing to launch the first product, and here it's not the MRI traceable polymer, but it's the stealth hydrophilic PPE for stealth applications, such as prolonging nanoparticle circulation time, etc., etc. We are not launching the product yet, but we would like to generally know which products might be interesting for community. So I have here a QR code. It will also show up on the next slide. You will get a questionnaire. It says a quote request, but I mean, it's completely not binding, and you do not really need to get a quote. So if you think that this product might be interesting for you, but you don't want to buy, then you can still cross it out and then just write this option that you don't need a quote, because for us it would generally be good to know which, if these polymers are anyhow interesting for research community. And I think they are, because Frederick published a few papers on this, but there are still many unknowns which could be published and which could be a nice student or PhD student project. But yeah, I would like to do, let's say, this sort of customer validation and customer research. So if you have time, it would be nice if you could scan. Yeah, finally, I would like to thank you for listening. And was actually, yeah, almost first time for me doing this lecture, so I'm curious how it arrived. I would like also to thank Timo, who did most of the synthetic work, Lena also worked, and obviously Frederick, the chair of the Sustainable Polymer Chemistry group. Originally, I moved there because I thought I will start a research group there, but yeah, it became a company. Our founders and yeah, again, collaborators, and yeah, you for being here, and yeah, the organizers for the opportunity to present in this online conference.